Hi, my name is Sydney Shaffer, and I work in the lab of Arjun at the University of Pennsylvania. I will be telling you about our recent project looking at rare cell variability and reprogramming as a path to resistance to targeted therapy in melanoma. Melanoma highlights the promise and the pitfalls of targeted therapy, where patients undergo dramatic remission of their metastatic disease, as you can see here, but ultimately fail to be cured due to the emergence of drug resistance. This resistance process is thought to commonly occur due to Darwinian selection, in which the resistant cells are the only ones that survive drug following treatment. Patient-derived cell lines can serve as a platform for studying the resistance process. Here we have cells cultured in a fractional killing dose of vemurafenib, and after about 3 weeks, we see these resistant colonies that appear on the plate. We wanted to know if these resistant colonies were arising from one or from many cells. Thus, we used long-term time-lapse imaging of an entire culture dish. Since we did not know a priori where the resistant colonies would arise on the dish, we had to image over the entire area of the dish and then go to the final frame and find the resistant colonies and then track backwards to capture the resistance process. Here is one of the videos that we generated from this experiment. We allowed the cells to proliferate normally for a few divisions prior to the addition of the drug. Now let's track these two cells here as labeled by the white arrows. They just came from the same parent cell, so they're both sister cells. As we go forward, what you'll see is that the cell from above went up to form the two cells labeled by the green arrows, and they appear rather healthy in response to drug. However, the cell below, labeled by the white arrow, appears stressed. As the video continues, the cell labeled by the white arrow continues to be stressed and does not proliferate. However, the cells labeled by the green arrows end up forming a large resistant colony. We found this result perplexing, as two sister cells should have the same mutations and thus have the same potential to become resistant, unless the mutation happened during that last final division, which seems unlikely. This led us to think about alternative models for resistance. We considered two models for resistance. First, a genetic mutation model where resistant cells cannot revert backwards to being non-resistant. And two, a non-genetic variability model where cells have the ability to transition back and forth between non-resistant and a pre-resistant state. We tested these models using the fluctuation analysis experiments of Luria and Delbrück. We generated single-cell derived cultures, then added vemurafenib and counted the number of resistant colonies in each culture. If resistance were heritable, we should see occasional cultures with very large numbers of resistance colonies due to mutations that occurred early in the expansion process. However, if resistance is not heritable, then all the cells in the culture should have equal likelihood of being resistant. When we count the number of resistant colonies, we can find differences in the distributions for these two models, as shown by the simulated data on the side of the slide. Surprisingly, we found that our experimental distributions were not consistent with a heritable phenotype, and we confirmed this using simulations and significance testing. See the paper for more details. Thus, we proceeded with this transient model of resistance, where the green cells are pre-resistant ones, and they can transition back and forth between non-resistant and pre-resistant. This led us to the question, what are these pre-resistant cells and what is unique to them? Specifically, are there gene expression differences that are responsible for this pre-resistant state? To tackle this question, we started with determining what genes are responsible for drug resistance. We performed RNA sequencing on clonal isolates prior to drug, after 48 hours in drug, and upon the development of resistance. And from this, we identified a set of genes that are uniquely upregulated in drug resistance. While these genes all have low expression in the untreated samples by bulk RNA sequencing, we wondered if there was a rare cell population that already had expression of these markers, and if these were associated with the pre-resistant state. Thus, we next performed single-cell analysis using RNA-FISH for these resistance marker genes. With high-throughput imaging platforms designed to image thousands of cells, we found a rare population of cells with high expression of the resistance genes. Here's an example with AXL. In this image, each spot is an individual molecule of RNA, and this cell on the left has very high levels. However, the cell next to it has no expression, and most of the cells on this dish have no or very low expression. Looking across the entire data set, we can find these high-expressing cells scattered throughout. On this plot, each dot is a cell, and the number of RNA molecules is indicated by the color of the dot. The dark blue dots represent negative cells. We also developed methods for iterative hybridization to allow us to multiplex the RNA fish assay and perform this for 19 different genes in the same cells. From these experiments, we find that this rare cell behavior occurs for many genes in the WM989 cells, as shown by the histograms here. However, we also find that this rare cell expression occurs in other melanoma cell lines, other cancer lines, primary melanocytes, and patient-derived xenograft models. See our full paper for details. Furthermore, we find an even rarer population of cells that expresses multiple of these resistance marker genes, and the frequency of these cells is close to the expected number of resistant colonies on the dish. Next, we wanted to know if these rare cells were actually the same ones that develop into resistant colonies. Fortunately, two of our resistance marker genes are also cell surface markers, EGFR and NGFR, so we used fluorescence-activated cell sorting to isolate the high fractions of these cells and applied drug to them. After 2 weeks, we see many more resistant colonies coming from our rare EGFR high and NGFR high populations, but the most resistant colonies come from the population of cells that are positive for both of the markers, confirming that these rare cells with high expression are the same population that develop into the resistant colonies. This led us to our next question. If the pre-resistant state is transient, then how does it later become permanently resistant? We hypothesized that resistance occurs in two phases. First, rare cells become transiently pre-resistant, then adding the drug initiates a cellular reprogramming or a burning-in process in which the stable resistant phenotype is obtained. To determine whether adding drug led to reprogramming of pre-resistant cells, we isolated these cells before adding drug as well as at 1 week and 4 weeks in drug. We performed RNA sequencing and found that the pre-resistant cells express only a very small fraction of the resistance markers. However, after adding drug, the percentage of the resistance genes expressed increases, with almost half of the genes expressed at week 1 and about 2/3 expressed at full levels by week 4. Thus, we show that with time and drug, the resistant cells undergo a transcriptional reprogramming to take on this permanently resistant state. We performed a similar analysis using ATAC sequencing on these same samples as well. See the full paper for details of that analysis. Together, our results show that when one looks at resistance at the single cell level, what initially appears to be an example of Darwinian selection is actually more complex. Our work supports a multi-stage model for resistance that includes a pre-drug transient phase followed by a drug-induced reprogramming phase. Thank you for watching our slidecast. See our paper for more information.